Distinguished Professor Emerita Sharon Strauss graduated from Harvard University cum laude in 1978. In 1984, she received a master's degree in ecology and a minor in statistics from the University of Minnesota. Dr. Strauss completed her PhD studies in 1988 at Florida State University.
After her postdoctoral training in the departments of entomology here at UC Davis and the University of Illinois at Urbana-Champaign, she joined the UC Davis faculty in 1994. Here at UC Davis, Dr. Strauss rose to the ranks of assistant, associate, full professor, and distinguished professor. She served as the ecology program officer at the National Science Foundation from 2001 to 2002. Dr. Strauss chaired the Department of Evolution and Ecology from 2013 to 2016. Over the years, Distinguished Professor Emerita Sharon Strauss has been a strong supporter of women and underrepresented minorities. Dr. Strauss's research has been cited more than 17,000 times and has an outstanding H-index of 63. Among many accolades, Dr. Strauss has been elected as Fellow of the California Academy of Sciences in 2009, Senior Fellow of the Ecological Society of America in 2015, and Fellow of the American Academy of Arts and Sciences 2015.
Last year, Dr. Strauss was elected to the National Academy of Sciences, to which she'll be inducted next week. Please join us in welcoming Distinguished Professor Emerita Sharon Strauss to the podium.
Thank you very much for the introduction and Walter for inviting me. And like Ralph, I still feel superfluous after hearing all of Barbara's amazing accomplishments. But I'm going to talk a little bit today about some of my research and great, thank you. So the title of my talk is Do Relatives Make Good Neighbors and Other Thoughts on Biodiversity. So I want to start by, oops, let me go this way first. Nope. Oh, all right. I'm missing that slide. I want to say thanks to the colleagues who were invited, who have contributed to the major portions of the research that I'm going to be talking about today. And I, at one point, had a slide of all of my students and postdocs that really have inspired me through the years and have enabled me to do the work that I have been able to accomplish. And also, I have to thank my colleagues in evolution and ecology and on campus generally, because they really push me and inspire me and encourage me to be the best scientist that I could be. So when we go into the natural world, it's hard not to be amazed by all the different forms and diversity of organisms that we see out there. And that was certainly something that impressed Darwin and was really a motivator for him to think about the evolution of life on Earth. And let's see. Whoops, sorry. Right. Okay. So one of the basis, and I would argue the major basis of this diversity is ecological specialization. So if every organism could perform every role, then we would have one plant, one bird, you know, that kind of thing. But what we see is that we have individual species that are specialized ecologically on particular functions. So here we have two finches. This finch can eat a variety of seeds, like this goldfinch, but it really can't get into these pine cones, whereas this related crossbill has this weird bill that is adapted for prying open the scales of these pine cones and other conifers and extracting the seeds. Similarly, there's a lag in this thing, sorry. Similarly, we have this side-billed hummingbird, and its beak is very well adapted for the curved flower in the Heliconius plant, and that is going to allow it to get into the nectar better than these straight billed hummingbirds, which are much more adapted to straight tubular flowers. And even plants have this kind of specialization. This is an amazing super bloom at the Carrizo Plain in 2017. And this is happening right now. It's a little less purple this year, but it's still absolutely amazing. So if you have some time to go down there and see it. We have what looks like this mosaic of plants. And what we're learning is that the different colors that we see are different species that are adapted to small differences in soil chemistry, and also to aspect like whether or not they're getting a lot of sun on a south-facing slope or less than a north-facing slope. And so all of these things are examples of specialization that lead to biodiversity. And so we think about specialization in terms of the ecological niche that species play. So what is a niche? It's basically the role that an organism plays within an ecological community and the conditions under which it can thrive. And so that would include the range of abiotic factors like climate that a species can tolerate, the resources in the ecosystem that a species uses. It could be soil nutrients, but it could also be other species. So it includes interactions with the biotic environment, that is other organisms. So disease, competition, and beneficial interactions even where species can help each other survive in particular environments. So we can characterize the ecological niche, and this is work that I'm actually doing right now with Annie Schmidt, Julian Malouf, and Jenny Bremer, who might be on the Zoom, where we're looking at the germination properties across a large group of native mustards that are called jewel flowers that I'll be talking about a little bit later. But here we've got different temperatures and water availabilities, and what you can see is that some species have a very narrow niche. The warmer the color, the better they like those conditions. And other species have a very wide niche. So we can start to characterize the ecological niche and aspects of it that different species play. And the other thing we understand is that niches evolve. So when there's a speciation event, when one species turns into two species, and there are a number of different mechanisms through which that happens, we often find that the niche of the two species is very similar initially right after speciation. And there's a term for this, it's called niche conservatism, which is the tendency of related species to retain niche traits through speciation events. So this suggests that very closely related species should occur in similar habitats and perform similar roles in ecological communities. So how do we define relatives in evolutionary biology? This is just a quick sidetrack for those of you who are not biologists. And that is basically the more similar the genetic code between two organisms, the more closely related they are. And that's just the same as, you know, we're more closely related to our brothers and sisters than we are to our cousins. And in this particular case here, we have the great apes that are part of a larger primate clade. And you can see that these two species, the chimpanzee and the bonobo, are both in the genus Pan, and they're our closest relatives. So these guys have diverged about 2.5 million years ago. And then the group of apes that we belong to, some of them are extinct now, right? But the extant ones, we diverged from this branch of the primates about 5 to 6 million years ago. So you can see that, you know, with longer and longer periods of time, since we were close relatives, you know, we see this way that we can build an evolutionary tree and characterize how closely related different species are. And Linnaeus actually did this before he knew about DNA, but he was sort of thinking about similarity and how we might group organisms in terms of their similarity. And he came up with this classification system for species. So here we have the red fox in the species Vulpes vulpes. Here we have other foxes that are in the genus, but not just the red fox. And you can see they look similar, but you know, we move up to the family, which is the family that dogs are into. But here's an example of the wolf, which is also in that family. And then above that, you can see we get more and more diverse forms. But you can see that through evolutionary time, there's still a lot more similarity when organisms are more closely related than when they're more distantly related. And it often follows these general, what we call ranks of species, genus, family, and then above family. And that's pertinent to my talk today. Come on. Okay. So Darwin had some ideas about what were the effects of similarity between species. And he wrote, as species of the same genus usually have much similarity in habits and constitution. Well, anyway, they should be found together in nature. If they're similar, they should be found together in nature. The other part of his thought, though, was the struggle will generally be more severe between them if they come into competition with each other than between the species of distinct genera. So here he's arguing that we shouldn't actually find them close together because they should exclude each other because they're scrabbling for the same resources and therefore they wouldn't be good neighbors. So coexistence between close relatives is going to reflect the conflicting forces of competition and niche conservatism or other things that limit coexistence. So I just want to show you some examples here. These are at the Bodega Bay Marine Reserve, which is a UC Davis reserve. And here we have our native goldfields, California, Lasthenia californica, and its very close relative, Lasthenia minor. You can see that they look really, really similar. They bloom at the same times. They both occur at Bodega Bay. They occur within meters of each other in some places. Similarly, we have these sand verbenas, again, both occurring at Bodega Bay. They look really similar. They live in similar sandy habitats, but they do differ in a few things here. So we have this, these very close relatives that at least at Bodega Bay seem to be in fairly close proximity. So what we wanted to do was actually test some of these ideas to measure ecological similarity of species and the strength of competition between them and how these translated into patterns of local coexistence in natural field communities. And then I want to take a step and go beyond competition and talk a little bit about other processes that could explain coexistence in close relatives and the patterns that we see. But first, I want to say that I am a huge animal lover. These are some of my favorite animals: pangolins, giraffes in the field. I mean, you've seen them in a zoo, but there's nothing like seeing a giraffe on the African plains to make you go, whoa, how did that organism ever evolve? Our son studies hyenas. I've learned a lot about hyenas. So even though a lot of people go, ooh, I hate hyenas, they're actually amazing too. And yet I don't study any of these amazing organisms that I am truly passionate about because I can really do a lot of experiments with plants. And they are so helpful because they don't run away. And so I can actually force them to live in the niche of another species, which you really cannot do for an animal. So we use the Oprah approach. We can take a species and we can plant it into the niche of another species in the field. And we can also know the relatedness of the planted species to the resident species. And then we can interview them and we can say, how did you grow and survive with that relative? And so then we can vary how closely related the resident species is to our transplanted focal species. And we can start to try and get at some of these ideas that Darwin proposed. So we're hoping to, what we're trying to do is, or what we did do, is combine experimental approaches with our knowledge of the evolutionary history of relatedness between the species. And taking into account that evolutionary relatedness will give us some more insights into which species we find in diverse communities. So we did this work at the Bodega Bay Marine Reserve, which is part of the UC Natural Reserve Systems, and it's run by UC Davis. It was occupied and treasured by the coastal Miwok tribe in the past. And what we have at Bodega Bay is a range of habitats. It's actually a small reserve. The scale here is 1 kilometer, but it does have the San Andreas Fault, which represents the junction of these two different plates, geological plates, which means that we have a variety of soil types. And we also have, we have these coastal bluffs that get a lot of salt spray that make the soil much saltier here. We have small wetlands. We have dune habitat and grasslands. So we have a large variety of habitats. And so we do have a large diversity of native plant species here. Over 200 native plant species occur in this small area. So we use that as the backdrop to answer these questions and to do our Oprah experiment. And so we did this field transplant, and this is our experimental design. We take a focal species, in this case it was Plantago erecta, and then we planted it into patches of plants that were either other individuals of Plantago erecta. So the neighbors were the same species. We call that con, which is the same con specific. We planted it into the habitats that another species that lived at Bodega that was in the same genus occupied. So that would be the congener here, Plantago subnuda, and then into another habitat that was occupied by a member that was in the same family, so Linaria canadensis, and finally, a much more distantly related family. This is a legume, this is a clover. That might have been one set of treatments. So that's one example. I just show you a couple more. Oh, we did this at 2 replicate. So we repeated this whole experiment at 2 replicate sites per treatment, and we had 15 species sets, and we also did this for 2 years, and we didn't even use the same species. So we used, this was a large experiment. And this is just showing you some of the other combinations that we did. So this is in the carrot family, Senicula crassicalis, transplanted into other patches of this species, into Senicula arctopoides, Daucus pusillus, all in the carrot family here. And then this is in the aster family, a thistle, a local thistle. Here we have 2 Camisoniopsis and Camisonia and Plantago erecta. So this is what our experiment looked like. And then rather than rely on those ranks of same genus, same family, distant, etc., we were actually able to build a more refined evolutionary tree that gave us a more accurate degree of relatedness, so that we could measure the number of the millions of years or thousands of years diverged that each species, that the focal species was to the resident where it was transplanted. So I'm just going to go over this at the axes and go over some of the expectations that we might expect based on Darwin's predictions. So here on the x-axis, we have the relatedness separating the transplanted species from the resident in millions of years. That's based on the phylogeny that I just showed you here. And then here we have some measure of performance or fitness. So how well did they do? So if they do well, they would be high here. And if they did poorly, they'd be low here. And if they were closely related to the resident, they would be here, and distantly related, they would be here. So if we think that more closely related species do worse in each other's niches, like for example, if they were competing very intensely, then we might expect this kind of relationship. Alternatively, if because they had similar preferences, because they were recently, they were not so long ago, they were the same species and they have diverged, then maybe we expect them to do well in the habitats of their close relatives and poorly in distant relatives' habitats. So let's take a look at what our experiment actually showed us. So our first measure of fitness is survival. We'll also talk about reproduction at the end. So here, what we found was that if we looked at just the initial survival of these guys, we find that we had similar survival of species up to about the family level. So this would be where the millions of years diverged of the confamilial treatment on average. And we see a modest decline in survival at distant relative sites. So we see that basically they tend to do better, at least initially, surviving in close relative sites relative to more distant relative sites. To understand the role of competition, I left out one major part of our experimental design was we actually removed the resident from half of the plots. So where we had, this is basically our control plot where we planted our transplanted species in, we had our resident species that was part of our experimental design. And then we had our neighbor reduction treatment where we actually removed this resident species. And then we could look at the performance, and that would be trying to get at the idea that Darwin had that maybe closely related species would actually compete more intensely with one another. And so here we have a metric of how intensely species, individuals compete with one another. And what you can see is if you score low here, you're highly suppressed by your neighbor. If you score high, you're helped by your neighbor. And again, here we have the I don't know why that's doing that. The relatedness of the transplanted species to the resident species. And so what we find is that when an individual is transplanted into either a conspecific member of the same species or a congener, this is a positive slope no matter whether or not we have the species include the species in the relationship, we find they do more poorly. They do in fact seem to be more negatively affected by the presence of that other individual. So they do appear to compete or somehow negatively interfere with each other more than with more distant relatives, as predicted by Darwin. And if we take a look at the combination of survival and competition, and we add it all up to look at the biomass of survivors, these were all annual species, so biomass is highly correlated with overall seed production and fitness. What we see is this sort of curvilinear relationship where species do well with other members of their own species and with distant relatives, but they do relatively poorly when they're surrounded by neighbors that are either in the same genus or the same family. So that would make us predict that we should see spatial separation of members of the same genus and members of the same family at Bodega Bay. So let's go see whether that's really true. We had these over 200 native forb species at Bodega Bay, and then we can put out quadrats all over the place, which is what we did, hundreds of quadrats. And then we actually looked at, well, what do we see? Do we see aggregation or dispersion when we do these quadrats? So inside this 1/4 meter quadrat, so that's not very big, there are 20 species from 19 different genera. So which is genera is like multiple genuses. There's no such thing as genuses, but that's what you call many species that are different genera. Okay, so we could calculate the probability of encountering a specific rank, so species, genus, family, or distant in the quadrat, and also as a function of the spatial separation of quadrats. And what we find is that the rates of co-occurrence with congeners, members of the same genus, were low, much, much less common than we would expect by chance. So when we generated our expectation by chance, we took into account how abundant that species was. If it was super abundant, maybe we'd expect to see it in lots of quadrats. So taking into account how abundant a species was, we could then generate the expectation of how likely would it be that these two species actually could be found together. And so what we found was that congeners, but not confamilials, are significantly spatially dispersed. Is the Zoom still working?
Yeah, this one's good.
Okay, I can just speak loud. Don't worry about it, right? You guys can still hear me? I'll speak loud. Okay, don't worry about it. Thank you. So the take home of our experiment was that congeners are more ecologically similar, survive better in each other's sites, and compete more strongly with each other. And that the net results of these conflicting forces was fine scale spatial separation of congeners at Bodega Bay. But what other forces might drive habitat separation in close relatives? So another force that I have increasingly been focusing on later, since that initial ecological experiment, is this process of reproductive interference. So reproductive interference is basically an interspecific interaction, an interaction between two species that are really good species. They're reproductively isolated. They do not hybridize. They do not make offspring together. But somehow, if these two different species mate together, one or both of them suffer reduced fitness. So let me just give you an example because that's easier to understand. Here we have two species of frogs. This is Rana temporaria and Rana dalmatina. And it turns out that if there's high density of Rana dalmatina, there's a lot of matings going on between males of this species and females of Rana temporaria. And then she produces a much higher proportion of inviable eggs as a result of that mating. So the habitat that had many of these species would really be not suitable for Rana temporaria, not because they don't like the habitat or that they compete more strongly, but because there's this reproductive interference that's severely reducing the fitness of these species. Luckily, this is a frog and she could maybe hop away if she didn't want to deal with that, and if she somehow had some awareness that this was not a good thing. It's a little bit harder for a plant. They're sessile. They're stuck in the ground. They cannot move. This is why I like to work with them. But if they look alike and they bloom at the same time and they share a pollinator, well, how do plants mate? Well, they actually rely on pollinators, many of them, to take pollen from one individual and move it to another individual. Well, if that pollinator instead moves it to another species or vice versa, there are opportunities for reproductive interference. So we could expect close relatives to experience reproductive interference if they reproduce at the same time, they look similar, they occupy the same or nearby habitats. And I argue that these criteria actually could be met by many close relatives, even at Bodega Bay. Even if congeners don't occur within a 1/4 meter quadrat, we still know there are congeners that are out at Bodega Bay, possibly because they share similar habitat preferences. So I just want to go over a case study of what forces prevent local, very fine scale, local coexistence of two California jewel flowers. And, hint, hint, it might entail reproductive interference. So this is Streptanthus breweri and Streptanthus hesperides. They're in a group of native mustards called the jewel flowers because they're actually really very beautiful flowers. And my colleagues and I have developed a phylogeny for this group, so we know how closely related these species are to one another. And so these two species are right here on the phylogeny, and they're very closely related. They're not sister species, but they're very closely related. And if you look at where they occur in California, they both have a fairly limited range, but this green blob here is the range of Streptanthus hesperides, which I'm just going to call hesperides, and it's nested within the breweri range. And if we go to finer and finer spatial scales, this is the McLaughlin Reserve. This is another amazing reserve from the UC Natural Reserve System that's also run by UC Davis. We can find, that they basically are occurring often separately at the reserve, but occasionally we can find them at sites where there's overlap. So when we take a look at these sites, so here, one of the interesting aspects of the ecology of these species is they like these strange serpentine soils. Serpentine soils are the ocean crust that have been uplifted. They have a really weird chemistry. They have a lot of nickel. They have low calcium and high magnesium, and they're stressful for many other species to grow on. They also have these very coarse soils, as you can see here. So we get these sort of more barren outcrops of serpentine soil nested within these more mesic sites that are invaded by a lot of our exotic grasses. But right here, we can get a lot of our native species because the exotic grasses can't handle it. And this is where Streptanthus breweri and hesperides hang out. And here on this outcrop, even though it looks like they're really growing right together, Streptanthus hesperides is occupying this part of the outcrop, and Streptanthus breweri is occupying that part of the outcrop. And very rarely do we see that. And Sarah Ashlock, who's right here, spent a lot of time with me out at this field site. So yeah, we almost never see this. You can't see them because their leaves are the same colors as the soil. And that's an adaptation to hide from herbivores, which is some other work that I've done. So You can't see it, but trust me, they're there. So what's limiting coexistence of these guys? Why are they seem to like the same outcrops, habitats. So what is limiting? So maybe the different parts of the outcrops or the different outcrops at McLaughlin have different soil chemistries. So we could take soil samples and we can use this sort of multivariate analysis to describe the chemical composition of these soil. And what we see is that basically reiterating the geographical distribution, we see that Hesperides occupies a narrower range of soil characteristics than does Streptanthus breweri, but it's completely nested within breweri. So breweri should be able to live anywhere Hesperides can live, and maybe Hesperides can't live everywhere where breweri can live, but there's still be a lot of overlap. So we decided to do a field transplant experiment. Again, they can't run away. We get to ask them questions. And then we can also look at how closely they compete to see whether one of them is excluding the other one. And then we're also going to look at reproductive interference. And this was work done by Kyle Christie, my PhD student. This was a huge experiment. We had 6 paired outcrops where we had both breweri and Hesperides at the same outcrop, but at different parts of it. And then we transplanted equal numbers of seeds into these quadrats. And then we looked at how well they grew and survived in these outcrops. So this is what we would expect if they were locally adapted to the conditions of their outcrops. So breweri seeds should do best in, breweri seeds should do best in breweri habitat and worse in Streptanthus Hesperides habitat or soil, and vice versa. Hesperides should do poorly in breweri habitat and better in their own. So what about germination? It turns out we find the exact opposite of germination. They actually do slightly better in the other one's soil. So that cannot explain why they don't co-occur together. So there's no evidence for local adaptation to small-scale soil differences in the germination of our transplants. If we look at competition in the greenhouse where we use soils, we did not find any. I'm not going to go over it, but all I can tell you is these results are identical, whether they're growing with their own species or the other species. It is not because one can outcompete the other. Okay, well, what about survival? Maybe there are some differences in the field, something to do with the water relations or soil textures or something like that. And what we do see here is that we do see an indication that Streptanthus Hesperides, remember that one has the narrower range, actually does do better on its own outcrop on average than it does on average at the breweri outcrops. In contrast, we don't see any difference between breweri in how they perform in their own part of the outcrop or in the Hesperides part of the outcrop. So this could explain why we might not see Hesperides in the breweri part, but it doesn't explain why we don't see breweri in the Hesperides part. And in fact, if we then look at these transplants, how high they grew, how much fruit they made, there's actually no difference between these guys, regardless of where they were planted. So why doesn't at least breweri occur in Hesperides sites? And none of these differences, which were significant, or even if there was a trend, they shouldn't result in the extreme spatial isolation that we see. We should see some of them there some of the time, and that's not what we see. So what are the other forces limiting coexistence? Well, I've already hinted that maybe reproductive interference is important. So it turns out these guys flower at the same time, and they share the major pollinator at the field site, which is this Bombus vosnesenskii, one of our native, really important bumblebees, and one of the most common ones. And here it is visiting a Streptanthus Hesperides plant. So part of Kyle's thesis was characterizing reproductive isolation. That is, when do species become good species, and how far along the speciation process are they? By crossing a lot of these species of jewel flowers with one another. So the ones that are in yellow are are much more closely related. The ones in red are much more distantly related. The ones on the diagonal are the same species. And then he could look at the production of viable seed. And what he found was that basically, if species were diverged at least 2 million years ago or longer, they were reproductively isolated. You put pollen of another species on that species and nothing happens. But for species that are more closely related, they're not so well reproductively isolated. They don't make viable hybrids, so they are good species, but they actually do start producing inviable seed, and that has to do with incompatibilities in the endosperm, which is a seed. You don't need to know that. Biologists might be interested. But anyway, they end up producing a lot of inviable seed. So we thought, well, we cannot figure out this mystery. We should go back to our plots and we should see how many individuals have survived in these plots and how many of the immigrants individuals are present. So when we planted them in equal frequencies within these plots, but the outcrop was dominated by one or the other species. So if this is the Hesperides outcrop here, this one here, then you can see that there's fewer bruri because all of these plants are Hesperides, right? So if a pollinator is moving around, the chances that this bruri is going to get Hesperides pollen is really, really high. So we could say, well, maybe if reproductive interference is important, then the ones where there were more bruri that survived in a very local neighborhood, they might have higher seed than the ones where there wasn't very many bruri left to mate with. So we took advantage of these different densities of surviving transplants to assess the possibility of reproductive interference. And this is what we found when there were more of the same immigrant species in the plot. The seed viability that those individuals produced was higher. And in fact, if we look at this, seed viability patterns had the greatest impact on relative fitness on the outcrops. So if we look at seed viability in our field transplant experiment, what we see is that bruri produces way more viable seed when it's on its own outcrop than when it's on the Hesperides outcrop. And vice versa, Hesperides produces less viable seed when it's on the bruri outcrop and much more viable seed when it's in its own outcrop. And we're only talking about the migrants in those little squares. And when we repeat this in the greenhouse, in the same soils that we took from the same outcrops, we don't see any effect on seed viability, which is the smoking gun that it's actually the bees that are moving the wrong pollen that's causing the seed inviability that we know occurs from Kyle's crossing experiments. So reproductive interference is the most parsimonious hypothesis to explain why these closely related species that seem to share their same soil preferences, that seem to grow as well, survive just about as well, why they don't co-occupy these habitats in the field. So to conclude, close relatives do share ecological and niche traits, more so than more distantly related species. So we have to give Darwin a check mark on that one. And close relatives do compete more intensely. Yep, he was right. And they don't co-occur. And it might be because of that, but I would like to argue that it may be because recently diverse species may also interfere with each other reproductively. So a different mechanism from some of the ones that Darwin proposed in The Origin of Species. So I'm going to tell you that close relatives generally do not make good, at least immediate neighbors. Bear that in mind when your brother wants to move next door. But I feel like some of the insights that we got were based on our approach that we took, because we could combine our experimental and contemporary approaches with our historical knowledge of the long-term evolutionary history of these species to more deeply understand which species we find together in natural communities. And with that, I have to highlight Brian Aneker, who did a lot of the Bodega Bay work of transplanting those into all those resident sites. Eva Lou developed the phylogeny of the jewel flowers. Kyle did all the amazing work on the last part of the project. And my current collaborators with whom we're characterizing the ecological niche of the jewel flowers in general, especially with respect to germination. And thank you all for being here and listening.
Thank you, Sharon. Thank you. Thank you very much. Could you switch the camera? So before you leave, make sure that you sign the poster for Barbara. And to close in this event today, I would like to call Peter Wainwright. He's the chair of EVE, and he's going to say a few words, and then we can go home. Thank you very much. Thank you all for attending. Peter, thank you.
EVE is evolution and ecology, and many of us in EVE love organisms, birds, fish, flowers, bees, you name it, even Drosophila. If you've ever been to the Bodega Marine Lab, it's an amazing place as a marine scientist, just an access point into the fantastic coastal fauna and flora of Northern California, which is an amazing place. But honestly, the hillsides around the Bodega Marine Lab are probably even more astounding this time of year and further into the spring and other times of year because of the amazing plant diversity there. And I know over the years, my view of that landscape has changed remarkably because of Sharon's research. I no longer just sort of see it as this just sort of fuzzy mosaic of plants out there. I love the idea that there's so much diversity there and that there's organizing principles at work, even if it took so many hard experiments to sort of work them out. Now, I want to close by telling you a little bit about Sharon that she didn't tell you, and that is what an amazing faculty colleague that she was and still is. She hasn't been retired for very long. But honestly, I don't think I can think of a sort of single colleague who so embodies some of the most, I think, important sort of qualities that you see in good fellow faculty members. She really cared about students. She was very creative and thoughtful about making opportunities for undergraduates to get involved in research, and also for stimulating their interests in sort of parallel or related areas. Just very effective that way. Very thoughtful and caring and just clever with the work that she did with graduate students, her own graduate students and other students in our program. And in a similar way with postdocs. She was really good at bringing along nurturing young careers in science. And as you saw, she's just wildly enthusiastic and energetic about her own ideas and sort of taking off with research and just getting insights into things that 20 years ago, we didn't understand any of what she just sort of told us about those systems and really figuring things out and doing nice experiments. And as chair of the department, I've also come to appreciate maybe one of the least appreciated wonderful things about Sharon, that she turns out that she's really good at retiring. She retired, I won't go into details on this, but she retired like a real pro, like she knew exactly what she was doing. So when it came time to do that, she did a great job of it. So thank you all for coming. And Sharon, it's been a pleasure. And I hope to see you give a talk on hyenas sometime soon.
Thank you, Peter. Don't forget to sign the poster. And also, if you want to recycle this name tag, just remove the part inside because you need this QR code on the back. This for an evaluation and for suggestion on how you should conduct this series next year. Thank you very much. Please keep that for the scan code to scan and tell us what is your opinion about that. Okay. Thank you very much. And we are done for today.